previously said, we're talking about nutrient loading and runoff, and maybe there's a couple slides about algae blooms, and one of them might be the title slide because it was a really cool picture. We're going to be talking mostly about kind of the things leading up to that algae blooms, in all honesty. So giving a brief kind of rundown on eutrophication and nutrient loading, and then we'll be talking about one of the studies that we've been doing at work over in Spring Lake. Part of what our lab does, in addition to being part of the university, is our lab. I work for Dr. Al Steinman, who's the director of the institute out in Muskegon. And so in addition to helping grad students with their projects, we also do work kind of like as environmental consultants for different watershed groups in the area. And we have been working with the Spring Lake Board, doing some monitoring for them. So that study fits in really nicely with what the topic today was about. And then after that, we'll head outside, and if you want, you can get your feet wet, but it looks pretty mucky, so I don't know if I'd recommend it. I have waders, so I'll be saved. So as you can see here, just to start off, I've got two different pictures of two different lakes. You can see that there's some obvious differences between the two. Water clarity is pretty key there. One of them has a lot more algae. One of them you can kind of assume has different types of fish and other organisms living in either lake. And so ecologists differentiate between these two different kinds of bodies of water with two big broad categories called a trophic level, which is kind of referring to the production going on, primary production for each area. So we have oligotrophic, low nutrient, low productivity kinds of bodies of water on the left-hand side, and high nutrient, high productivity areas that are called eutrophic on the right-hand side. Oops, I'm on the wrong one. There we go. So there's some other differences there. Sediment being more rocky or sandy, and oligotrophic, more carbon-rich, more mucky on the right-hand side for eutrophic waters. So this is a process that happens naturally. It's not necessarily a bad thing. It can happen on a geological timescale. Lakes naturally do fill in over time, over hundreds of thousands of years, as you see in this diagram, going from top to bottom. So top being oligotrophic, maybe just a recently scooped out glacial lake filling in over time with sediment being washed in from upstream or from uphill, plants accumulating and dying and building up organic matter over time. And then finally, at some point, it might fill in and just become a wetland or eventually a forest, things like that. But we do have what's called anthropogenic eutrophication, man-made cultural eutrophication, and we kind of speed up this process. And so that speeds up eutrophication from a geologic timescale to more of a decades kind of timescale. Things like runoff and erosion and discharge from residential, commercial, industrial, agricultural, lots of different sources, right, where things can be added either more sediment or more nutrients of various types or other chemicals. Pretty big in the news. PFAS has been a big one that doesn't necessarily help product production, but it's a source of extra chemicals and nutrients going in the water. So if you're not familiar with this concept, in this, what's going on in this picture, this is all part of a watershed. So I know that you probably have heard this term a lot recently. You probably already know all this, but just to make sure we're all on the same page, watersheds, this is all happening inside of an area that all drains to the same area. So if we're talking about where we're at right now, on the top right, we're in the Grand River watershed. All this flows down to Lake Michigan, so we're in the Lake Michigan Basin. So the watershed on the left-hand side, the general example there, is just one big catchment. Maybe it's surrounded by hills or other natural features on one side. All these rivers and lakes all kind of generally flowing to one outsource area. So if you have pollution or nutrient enrichment happening in one area there, it's all going to eventually flow down to the next area further downstream in that watershed. So another great visual example of what a watershed is and how it works can be found in this outreach tool called an Enviroscape. We've got one at the Institute that Dr. Vail uses for different outreach events. And what's really cool about this is it's a really great visual way to show pollution around the landscape, maybe fertilizer up on lawns or industrial output on the top right, the factory there. And then when the kids get to spray water, they make it rain, and all those nutrients wash downhill. They end up in the pond down at the bottom. Another great example that's going on here is point source and non-point source pollution. So the point source would be specific buildings and land uses that are easy to point to, but the non-point source pollution would be more broad things happening over a wide area that might be harder to target in particular. So when we're looking at the stuff today, what exactly the factors are we going to care about for our eutrophication and point source pollution, nutrient loading? We're going to talk about four of them today. We're going to be talking about phosphorus and nitrogen, which are two nutrients, two elements that are vital to life, the algae that uses them, and then also sediment, because sediment sometimes has chemicals and nutrients stuck to it on its travel down through the watershed. So first of all, we're talking about phosphorus. When we study phosphorus and different phosphorus concentrations, we're usually looking at a particular compound called phosphate. It's essential to plants and animals. You find it in fertilizers. It used to be in laundry detergents until they become phosphate-free. So it's a very important chemical, but having too much in an area can be bad. The abbreviations that we use are TP for total phosphorus and SRP for soluble reactive phosphorus. And the difference there is TP is literally everything. If you grab a handful or a cupful of water rather from the creek outside, literally everything in that jar or cup, whatever, that's going to be your total phosphorus concentration. But if you wanted to know just the biologically available sample, you'd have to filter out all the bugs, all the algae to find out just what is dissolved in the water column. Okay, so the things that would be the SRP, you'd have to filter everything else out and just have a pure water sample. Whatever is dissolved and remaining in there, that's your SRP. And so those are two interesting things that we look at for our work. Phosphorus is usually a limiting nutrient in fresh water, and that means that there's so much demand for it that if you add any more to it, the algae usually goes gangbusters, tries to grow up and absorb as much as it can, and that kind of is the bottom-up limit on a food web. So normally you think of food webs being kind of interacting, something eats something else, but at the bottom of that you have algae and plants trying to gather up all the nutrients they can, fighting for sunlight, and phosphorus is a big limitation for that. So not only having the space, but also just the nutrients needed for life to happen. Phosphorus is also a sticky element. It can stick to dirt. So when you have maybe a mudslide or a rain event washing a lot of dirt from a farm field into the creeks that are going around it, the drainage ditches, a lot of that phosphorus is going to be washed in along with the dirt and can go further downstream much more easily. The second element that we look at is nitrogen. And nitrogen actually has two interesting chemical versions out in the world that we care about. Ammonia and nitrate, both of those are used in fertilizers also. Usually this is the limiting nutrient in saltwater systems or coastal systems, so not as usually limiting around this area, but sometimes it can be. And what makes nitrogen also kind of tricky for ecology is that there's some kinds of cyanobacteria, or blue-green algae is another term for them, that can fix nitrogen directly from the atmosphere. If they've got the right kind of little microscopic cells attached to them, they can just have access to the atmosphere and take nitrogen, which is like 77% of what you breathe anyway, and turn that into what they need to survive. So they don't even need to have the fertilizer version of it, they can just take it from the air. So algae in particular are a very diverse group of organisms. It's not just one group, it's actually several groups that all kind of get lumped together. You've probably heard about algae before, you've seen it outside. Some of it's actually pretty under a microscope. On the bottom we've got things like diatoms that have little glass shells, houses. There's big colonial green things, that's volvox on the top. When we measure algae, there's a couple different ways you can measure it. There's going to be ways that you can go through and identify species by species and know things about them based on their different life histories and habitat preferences or nutrient preferences. I had a professor at college at Michigan State when I was undergrad, and he could look at a sample and say, oh, this algae only exists when there's a high iron concentration in your water, which means there's someone polluting iron upstream. And he could know from his taxonomic experience, what the certain things in the water sample meant for the ecosystem like that. Other things we can do are measure the biovolume of algae, knowing the different sizes of them and how many are on your slide, knowing different things like that. But much more commonly, we just measure the chlorophyll A concentration of the sample. Chlorophyll is the pigment that plants and algae use to generate energy out of sunlight. There's a couple different kinds of chlorophyll, so chlorophyll A is the most common, and that's what we use. We have a machine called a spectrophotometer, and it's as easy as processing your sample and then putting it in the machine. It shines a light through it, and it tells you a very precise reading of this many micrograms of chlorophyll per milliliter of your reading. There are also some algae that kind of go out of control. We call those harmful algal blooms, or HABs, HABs. So I don't really have too much about harmful algal blooms today so far, but sometimes when you have too many nutrients in the water, your algae gets out of control, and you end up with harmful algal blooms that might produce harmful toxins when they get up to a bloom concentration. And that big picture that I had on my title slide was a harmful algal bloom in Lake Erie a couple years back. You might have remembered that one. It shut down their water supply because the toxins were sucked up into the municipal system. That was an algae called microcystis. The microcystin makes is toxic to humans and people. So our last bit that we were talking about today is sediment, and I mentioned that sediment can carry phosphorus with it as it gets washed down into ecosystems. That picture right there is the outlet of Lake Makatawa in Holland, connecting to Lake Michigan there. So it has a sediment problem there from all the agriculture further up in their watershed, and it's part of their ongoing restoration efforts that we help monitor there, just to help increase best management practices and measure some wetlands in the area that they've been restoring to see if this is slowing down. So sediment can enter watersheds through runoff and erosion, and it has not only the impact of having physical effects, but also it can carry chemicals with it. It can have biological impacts on a water body. So some examples of the biological impacts would be if you had a really nice fish hatchery. Some fish prefer to have gravel beds to lay their eggs, but if it gets a bunch of muck washing down the river, it might cover up those gravel beds, silt over, and those fish won't want to spawn there anymore. They'll try to find somewhere else, and you might lose your fishery or degrade it in some respect. So kind of transitioning now into nutrient loading. The things that we've all been talking about so far, about where these nutrients are coming from, I've kind of all been using upstream examples, right? Something comes from something else upstream. So when we're talking about nutrient loading, we actually have two different kinds of loading methods. We have what's called external loading, which is something comes from something else upstream or around it. So it's input received from the watershed, whether that's runoff or stream flow. And then there's also the opposite, or the complementary version, I should say, which is internal loading. And that's the stuff that's already in your lake or inside your watershed, and that's being recycled from within. So in a lake example, if you're talking about the phosphorus that was being washed in with your sediment or just from other fertilizer coming in from upstream, once it's in your lake, it might not just disappear. It might not be used up by all the algae right away. And there could be situations where the phosphorus is kind of stuck at the bottom of the lake and then it gets resuspended by winds or waves or jet skis. And then that phosphorus kind of hangs out again for another season, sinks to the bottom, and continually gets resuspended without leaving the lake. So in addition to having to monitor external loading, it's also important to keep an eye on internal loading rates, because sometimes what's flushing into your system is not flushing out equally quickly. You can accumulate a lot more phosphorus than you think over time. One way to fight that kind of phosphorus internal loading, though, is to do what's called an alum treatment. Alum is the same kind of thing you find. It's an aluminum compound. You can find it in the grocery store in the baking section, along with like cinnamon and sage and everything else, just a tiny little pack of alum. If you apply that in a water body, alum is unique that it will try to grab onto all the phosphorus that it can, so it doesn't go floating back up and become available to the algae. So it's no longer biologically available. So what you can do is apply large amounts of alum to a lake that makes a consistent cap over different areas of the bottom of the lake. So whatever is down in the dirt stays trapped down in the dirt. And then if anything else settles on top of it, then it can try to stick to it and hold on to it from the top also. So it's not a permanent fix, but it is one way to kind of restart your internal loading process, or bring it back to zero is probably more accurate. So one such study that we did was taking place at the same time as an alum treatment on Spring Lake in Muskegon County in 2005. Previous to that, Spring Lake had one of the highest concentrations of phosphorus measured in western Michigan lakes. Between 1999 and 2002, the total phosphorus levels of Spring Lake averaged about 100 micrograms per liter. And as some context for that, 5 micrograms is about as low as we can reliably measure on our machine. So that's 20 times higher than our lowest. And the restoration goal for Muskegon Lake of what they're doing to try to clean up Muskegon Lake's long-term AOC delisting for the EPA, that goal is 30 micrograms per liter. So if it's up to 100, that's 3 times higher than like an EPA recommended guideline for a similar lake in the region. Then some of the higher readings from Spring Lake at that time was averaging 100, but up to 631 micrograms per liter. So very, very high concentrations of phosphorus. So as part of a multi-year monitoring effort, our lab measured a bunch of different physical, chemical, and biological factors at sites throughout Spring Lake in several different years. So in 2003, before the alum was applied, and then also after the alum was applied, we measured in 2006, 2010, and 2016. We had 4 different sites in the lake. And we measured a wide variety of features that are listed out here. I'm not going to read them all out. But we have several different tools for measuring this. We have what we call, it's a YSI is the brand name, but it's a multi-parameter sonde that you can set out, and it measures a whole bunch of those physical conditions for you. We grabbed water samples to grab the nutrients to measure back in the lab, and also chlorophyll samples and bug samples by taking big crane scoops, not big, but about the size of your hands. It's called a ponar grab. And then we also grabbed sediment cores for an experiment to measure the internal loading rate of the sediments that we grabbed. So here's our sites. We grabbed 4 sites throughout the length of the main body of Spring Lake. And then from each site, we grabbed those cores. We brought them back to the lab intact. And that's kind of the cartoon diagram going on the right-hand side here. So we had those intact sediment cores with overlying water from the site that we captured. And then we had some air bubbling through. One of them was oxygenated, and then the other set was not oxygenated. It was anoxic with nitrogen and carbon dioxide gas. We sampled water at regular intervals at the times listed there. So immediately when we brought it back, 12 hours later, 1 day, 2 day, 4 day, things like that. And we make sure that we are replacing that water with additional site water, so it's maintaining a constant level of water inside the tubes. And so as we measure the phosphorus over time, as we have this incubation going on in the lab, so we can measure how much phosphorus is coming out of the sediment because there's no more water going into. We know what the constant is that we're replacing it with. And so any changes that we see over time, we can calculate a change rate, and measure that internal loading. We also did an experiment called a sequential fractionation, and that's seeing what the phosphorus is stuck to. So dirt's made out of metals and organic matter, but we were doing a test to see what kind of metals were present and seeing what phosphorus was sticking to in particular. If there's a certain kind of source of metals that this lake is sticking to in particular, sometimes you have different treatments besides alum that might be more effective in the long term. So this is a lot. You don't have to read all of this. I just wanted to kind of give you a general idea of what's going on here. This is just one of the result slides for some of the parameters. Each cell here has 4 different numbers, which is confusing, but we have 4 different years that we sampled. So the red one highlighted is 2003, which is before sampling. And then 2016, our most recent year of sampling, is at black and bold at the bottom of each cell. And so just to kind of give you a general idea, we're measuring depth. There's 2 sites that are deeper. I'm sorry, 3 sites that are deeper. Site 4 is more shallow. That was at the top end of the watershed. We have temperature being pretty consistent. We've got Secchi depth changing over the years. Secchi depth is a measure of water clarity, and so that is something that's changing over time. And one thing that I wanted to point out in particular is that those 2 sites, sites 1 and 2, are the deepest of our sites, and that helps go along with some of the patterns that we see later on for the nutrients. Again, a crazy huge slide. Don't try to read everything. The parameters that we have here, DO is dissolved oxygen, CHL is chlorophyll. We have, again, our 2 kinds of phosphorus, SRP, which is just the soluble, biologically available phosphorus, and then TP, which is total phosphorus, and pH is a measure of acidity or alkalinity in the water. So the things to look for here, I want to point out, sites 1 and 2 were our deepest 2 sites before. They have really, really, really low oxygen levels. And that is important for our phosphorus flux because having that low DO makes it easier for phosphorus to come back out, have that internal loading cycle happening. So if we have low DO, the phosphorus is more likely to come back out. And sure enough, if you look at the phosphorus concentrations, this is in units of milligrams per liter. The numbers I gave you before were micrograms per liter. So if it was 100 micrograms per liter, that is 1,000 times smaller than one of those numbers that are circled there. So a milligram per liter is going to be equal to 100, I messed that number up, didn't I? 175 micrograms per liter. The site 2 on the bottom there is almost 1 milligram per liter, and that would be 1,000 micrograms per liter. So these numbers are very, very high. And then if we look at some of our other stuff, I mentioned that we grabbed some invertebrates. Just to kind of show what's happening here, this is a measurement that we wanted to know because if you're putting a chemical that's sealing a cap over the majority of a lake, that's probably going to have some effect on the wildlife in the lake, right? So we made a measurement of the different counts of invertebrates, the different bugs that we could find of 4 big classes. And as you can see, between 2004 and 2006, that's when the alum cap went on, and there was a big dive in the number of invertebrates that we caught. So it was unfortunate, but we do see them that they come back. They start to recover after that first year that we sampled it. And they kind of leveled out, increased a little bit more in 2016, not quite as high as they were in 2004. But that does mean that there's bugs crawling around in the alum, helping out with maybe stirring up more dirt again. So that alum cap isn't permanent. It's not like concrete. It's a porous stuff. It's like sand almost. So things can crawl around in it and make holes. Anchors can drop into it. So that's just one of the clues that this is not a permanent long-term solution to solving your phosphorus problems if the bugs can help stir things up, right? So the numbers here are just statistical results, kind of pointing out the apparent total phosphorus release rates. So these are things that we were calculating based on our cores we had in the lab. And what we were seeing is that predominantly the total phosphorus release rates were highest before the alum was applied. After the alum was applied, phosphorus was a lot lower, and it stays low, which is good. That's encouraging. That means the alum is working, but it's not quite as effective as you might hope. It's not 100% effective. There still is phosphorus being cycled around, and that's kind of expected because there's still more phosphorus in the system that's settled on top of the alum. So that was the comparison of 2016, our most recent year, to 2003, which is our first before alum year. Just another quick recap of what we were showing. We mentioned measuring metals earlier. So these are just the different metals in our sediment samples. And we found out that calcium is far and above the highest concentration compared to everything else. And that means, I'm forgetting what that means, to be honest. But that means something important that I just cannot remember for the life of me. I did not have it in my notes. I think it means that alum was a good match for it, I think. I might be wrong on that one. I apologize. So kind of summarizing some of the points from before. Scrolling down. Maybe I do have it in my notes. Nope, just that calcium is the most abundant metal at every site. Okay. So kind of recapping, the results from our 2016, our most recent study, provided some partial evidence that the Spring Lake alum treatment is starting to lose its effectiveness. We saw in the big table that we had those really big concentrations of phosphorus down towards the bottom. And that's kind of surprising because even though we have alum still there, the bugs are still low, we didn't calculate a very high phosphorus release rate, but there is still a really high concentration of phosphorus down at those really deep sites. So that's probably related to the low dissolved oxygen concentration. The low oxygen is allowing the chemistry to recycle and come back up. It's not holding on to the phosphorus as tightly as we'd hope it would. The strongest evidence that I just talked about that was the SRP and TP data. Was higher than even the pre-alum concentrations. So there's been several studies before that are cited here that have all shown that chemical inactivation treatments, which is what alum is, it's inactivating the phosphorus, usually have short-term benefits, usually less than 10 years, or 4 to 20 years rather. And there's a variety of factors that can all rely on. So the shape of the lake, the amount of alum you can actually put in. Bioturbation is things like turtles or bugs or whatever digging up your alum, moving it around after it's been applied. Macrophytes are aquatic plants. The water column pH might be a factor. If you have a very acidic, a very alkaline lake, they would react differently to adding all this alum. The amount of sediment coming into your lake could cover up the alum treatment before it has a chance to really do anything. And you just have more phosphorus sitting on top of it anyway. But the big one on the bottom there is the external load reduction after alum treatment. If you seal off your lake, you have a nice alum cover on your lake, and there's just tons of sediment still pouring in with lots of phosphorus in there, then what have you done? Not much. It's just going to start right back over again. So the analogy here is like if you've got a leaky sink in your kitchen that's overflowing and spilling everywhere, and your basement's flooding, you should probably turn off the sink first before you call the plumber, right? You have to solve the problem before you start cleaning up. Otherwise, you're just going to have more mess later on. So as a follow-up from that, we started a new project that we've been doing over the past year about an external loading study. And this, I don't have very many slides on this because we haven't finished this report yet. I can't share data because it hasn't been finalized and approved. But over the past year, from last summer to this summer, we did an external loading study by monitoring these 7 sites throughout the Spring Lake watershed. And what we did was we were taking not only nutrient samples from these creeks once a month, we were also doing stream flow measurements. So the idea there is that we can measure how fast the stream is going, the concentration of nutrients, and that gives us a way to calculate the load of chemicals moving through at any given time. And we also were able to use some on-site pressure loggers to create hydrologic models, basically saying how much stream flow is going year-round. So every 10 minutes it takes a sample of water pressure. We can use math to say, okay, that means there was 30 centimeters or 50 centimeters if there's a storm or things like that. And so knowing how much water is going through, pairing that up with the chemistry data, we can say over the course of a year, this much phosphorus has washed through this site. We have several sites throughout that area of the watershed, so we have an idea of, for the entire year, how much phosphorus, how much nitrogen was washing down from those areas and into the lake. So a way to measure not only the internal loading that we've already measured, and now we have the external loading to pair with it. And so that was the example of what we were going to work on today. Going outside, I've got 2 pairs of waders if somebody wants to hop in with me. We've got the flow rod and a little box that measures flow, and it's going to be as easy as setting up a transect. I've got some worksheets if you'd like to have a copy of that, and you can kind of follow along and see what we're doing. The basic idea behind the math is that you set up a meter tape going across the width of your creek or your river, whatever the case may be, and you divide it into equal segments as shown there. And then once you have those equal segments, you have to measure the flow at each one of them. So whether you're doing a fancy machine like we have today, or if you're doing something as simple as dropping a ball in and seeing how fast that goes down, as long as you get a flow measurement for each part, each section of that creek, you could calculate flow for the entire creek for that transect. So the idea is that if you measure the depth, you measure the flow, and you have these standardized distances across, you can calculate just using geometry, the area of that particular chunk, the area of that chunk, the flow for each one. You've got a 3-dimensional number at that point for velocity, meters cubed per second or feet cubed per second, whatever you want to use like that. And then if you were to take a chemistry sample, you could probably just use 1 for the entire creek. That's what we do. You would be able to say, okay, my class went out, my group went out, we measured the flow of this area, we've got a water sample, and we know that this is the nutrient load for this day at this time. And that's what we do. That's we go out, we do that at base flow conditions. So that's when there's no rain for the past 3 days or so. The creek should be at its normal level. And then if there's also a storm, we go out. I don't recommend that because we're standing with a metal rod out in the creek during a lightning storm. Bad idea. Wait for lightning to stop. But if you want to do that, you would get a higher flow measurement and get higher nutrient readings most likely. So does anybody have any questions? I feel like I went fast.